Acho que agora já tá gravando. Se você quiser, você pode fazer a apresentação para a gente, Sandra, por favor.
Geralmente em português ou em inglês?
English,
In
please.
English. Good morning, everyone. A great pleasure to see you all. Today, I will present you Professor Ricardo Silvestre from Universidade do Minho, in Braga, Portugal. Ricardo have expertise in immunometabolism and macrophages and T cells and various infectious disease. He's a specialist in Leishmaniasis disease. And Ricardo coming to visit our department with fellowship from Cap Spring and doing classes in the post-graduation discipline. It's a great pleasure to have you here, Ricardo. Please.
Okay.
Can you share your screen?
Sorry. Okay, thank you. Thank you. Good morning to all. I think you can hear me well, right? So let me just start by thank you, Sandra and ICB for the invitation to be here. Also, CAPS for the fellowship and to spend these 15 days with you in São Paulo. So in terms of the presentation, I'm going to overview a little bit what you have done for the past 5 to 10 years on visceral leishmaniasis on a perspective, on immunometabolic perspective. So our lab in Braga, University of Minho, works with this model of visceral leishmaniasis, but so in an immunometabolic perspective. And so we have been dissecting several aspects of this network between Leishmania and the macrophages, and that's what I'm trying to focus here today. So I always like to start my talk by giving this slide, basically to introduce the metabolism, introduce the two sides of the coins that we should look to, and we are always trying to look at that. One of the things is that immune cells needs to reprogram their metabolism in order to do their function. So the concept of metabolic reprogramming is essential for understanding how immune cells cope with an infection or what other type of insult. The other part of the coin is, the other side of the coin basically is the environment. So the environment is going to take its toll on the immune cell. So while the immune cell needs to reprogram in order to do their functions, it has to cope as well with their metabolism. And those two perspectives are very important to understand these immunometabolic networks during an infection or other mechanism. The second point that I will always like to highlight is that each immune cell has its own metabolism. So we cannot mix metabolic interaction of neutrophils with macrophages or T cells. So in homeostasis and after homeostasis, they're going to cope with their environmental insults in a different metabolic way. So we have to basically what we try to do is try to understand the signals, these pathways that are modulated during the infection, identifying the sensors of nutrient sensors, for example, that all these mechanisms that lead to metabolic reprogramming in a certain condition, and try to understand what is the impact of that during the infection. That's what we're trying to do. So in a very generic way, and as a rule of thumb, so this was what has been done by different researchers and what has been shown is that if you want an inflammatory cell or an effector cell, normally these type of cells, either macrophage or T cell, for example, it depends mostly on a glycolytic metabolism, consuming a lot of glucose, transforming this glucose in pyruvate, and then fermenting the pyruvate to lactate. So it's a general mechanisms of glycolytic effector cells which are normally associated with a more pro-inflammatory phenotype. If you take a look to the other side, more anti-inflammatory cells, they're normally more oxidative. Whether they use glucose, glutamine, or lipids in terms of this for oxidative phosphorylation, it's still a matter of debate. But nevertheless, they shift their metabolism from more glycolytic to respiratory metabolism to cope with their needs. So this is generally a rule of thumb that we normally use on the immunometabolic field. But nevertheless, what is very important to take into consideration when talking about infection or, for example, cancer, is that these immune cells, when they get to the infected place, they found a metabolic restrictive environment. Let me just change. So this environment that they're going to find is going to limit their functions to deal with the pathogen or with the tumor cells or whatever the danger is. And this restriction is going to impact on cell fate and function. So it is very important. That's why I started to say not only the metabolic reprogramming is essential, but the environment that is going to find is crucial to understand, to fully understand the impact on cell fate and function. So the way that cell is going to be activated, producing cytokines, chemokines, and phagocytes, etc., etc. So we use this model, Visceral Leishmaniasis. So most of you, I'm sure, that know most of you know the disease. Basically, we use Visceral Leishmaniasis with always the two species that infect humans and animals that induce Visceral Leishmaniasis, namely Leishmania infans and Leishmania donovani. So you all know that this is a disease transmitted by a sandfly that bites and transmitted these small promastigote flagellated cells that is going to infect mostly macrophages, but also dendritic cells and other cells. And so we focus, because this is a disease that infects mostly the liver, the spleen, the bone marrow, and I'm going to show you as well the adipose tissue. So we focus on these type of organs, and most of the data that I'm going to present is basically on the spleen or on the liver. Why cannot change? Okay. In terms of immune response, so when you think about trying to understand this immunometabolism during infection and try to see which are the nodes that later we can pinpoint to try to a new therapy, for example, we have to understand how the immune response acts against Leishmaniasis. So this is a typical intracellular parasite. So what we want to induce in order to have elimination of the disease is a T cell response producing interferon gamma and TNF. So Th1 classical Th1 cellular response, as well as CD8 cytotoxic response that are going to activate macrophages and are going to kill the pathogen inside. When you talk about macrophages, so normally these M1 and M2 do not really exist in vivo, but we are trying to push the macrophage towards a more M1-like macrophage, meaning that inflammatory macrophage that produce TNF, IL-6, and other inflammatory cytokines, but mostly that produce a lot of nitric oxide and reactive oxygen species to eliminate the pathogen. On the other side, and talking only about Visceral Leishmaniasis, what we want to not to have basically is IL-10. IL-10 is the most immunosuppressive cytokine in this context that is associated with a susceptibility and growth of the parasite. So in order to try to combine these two notions, from one side, the immune response that we want, and the second, what is the metabolism that you want to occur in order to have a perfect world in terms of elimination of the parasite. So we started like 10 years ago trying to understand this metabolic crosstalk between the pathogen, between Leishmania and the host, and focus mostly on the main host, which is the macrophages. And secondly, identifying the pathways and the players that are involved that are involved in controlling the immune response and the pathogen maintenance. So we started, before that, this is the classical picture of, let's say, M1 pro-inflammatory. We just increase this. Well, this M1 pro-inflammatory macrophages. So the previous works by Luke O'Neill and others have clearly show that these macrophages, pro-inflammatory, that are linked with pathogen elimination are more glycolytic. So basically what was described is glycolysis is essential in the function of these macrophages. They produce a lot of lactate, so acidifying the medium. Basically they do that in the presence or absence of oxygen. They use this pathway to feed the pentose phosphate pathways as well. And they are characterized, as I said, by the presence of high levels of ROS, NO, and pro-inflammatory cytokines. On the other hand, the anti-inflammatory that I mentioned before normally is associated with this respiration, with this phosphorylative oxidation. So we started to take a look to the first periods of infection. So we took promastigotes, we took bone marrow macrophages. Normally we use murine macrophages, some cases human, but mostly murine. And we infect these macrophages with promastigotes, and we look to the first hours of infection, what happens. And basically, in a work that we have done with Thomas Laska and Marek Ohms from the German University of Leipzig, what we observed basically was that we had a signature, performing RNA sequencing at 6 hours after infection, and this was done with neutrophils and macrophages as well. We have a signature of several genes associated with high glycolytic pathways, so glycolytic genes. So we have an increase of this glycolysis upon infection, something that we can describe as innate because it's not specific to Leishmania at all. But nevertheless, wanted to understand what is the role of this glycolysis in the first hours of infection. So this is a seahorse graph showing that, in fact, infected cells that are in the red have more ECAR, so more acidification of the media, so are more glycolytic, as you see in this graphic. And basically, if we inhibit this glycolysis, so if we use 2DG, which is a competitive inhibitor of the first step of the glycolytic pathway, so it competes with glucose for hexokinase. What we observed was that inhibition of glycolysis after infection completely abolished the production of ROS, as you can see in this graphic. Not only it affects the reactive oxygen species production by macrophages, but also stops in pro-inflammatory cytokines. So all of this shows that, in fact, glycolysis in the beginning of infection is very important for controlling the parasite. So we did some in vivo experiments that show exactly this. So if we inhibit in the first hours of infection, or even previous to infection, glycolysis with 2DG, and this is, of course, in vivo, we increase the number of parasites. So we increase the susceptibility to infection. So this clearly shown in very simple terms that the glycolytic metabolism is crucial in the very early steps of the infection, and blocking that it is going to increase the survival of the pathogen. So we focus several studies in the beginning of when we focus on immunometabolism on these first hours of infection. So after mentioning the glycolysis and fermentation was very important, one of the things that we observed was that this glycolytic burst do not lasted for days. It was mostly confined in the first hours of infection, and then it lowers down. One of the things that we observe at this moment during this time period, and we are focusing on the first day of interaction, was basically that the cells, the infected macrophages, so they have an energy deficit very early after infection. So we measure the ratio of AMP, ATP. So the higher the ratio, the lower the energy capacity of the cell. And we observed that there was an increase of this ratio on between 10 to 14 hours after infection. It is specific with live pathogens, and it does not occur with irradiated or heat-killed pathogens. So it's specific for a viable infection. And this is recovered. And so in a very simplistic way, what we observe here in this paper was that, in fact, early after infection, we had this increased fermentation, but rapidly, don't forget that the increased fermentation is associated with inflammatory macrophages. So macrophages that are capable of killing the pathogen. So very rapidly, there is a metabolic switch that is dependent on the viability of the pathogen that transform fermentative or glycolytic macrophages in more equipped mitochondrial respirative macrophages. So these macrophages, which metabolism are more associated with an anti-inflammatory M2-like pathway. So how this metabolic switch is done, like I said, we knew that this was dependent on the pathogen. So one of the things that we thought of was AMPK, because AMPK is an energy sensor, which normally is associated, is activated when this ratio, AMP-ATP, is increased. So we know that AMPK is a quite conserved protein throughout even promastigotes, but special prokaryotes, but also, but particularly on eukaryote cells. It's a very conserved protein that has basically two functions. It activates all catabolic pathways, so the cell is an energy deficit, and activating AMPK is going to induce the activation of all catabolic pathways in order to produce energy. And it's going to shut down all of anabolic pathways to stop spending this energy. So what we observe basically, AMPK can be activated by this liver kinase B1, or it can be activated by this CAMKK. There are two proteins that are activated by two different pathways. I'm going to make this long story short. What we observed basically was that in our case, AMPK was activated by liver kinase B1 that is going to phosphorylate the threonine 172. So what we observed, in fact, was AMPK is activated during those time periods where we have energy deficits, and through their activation is going to increase mitochondrial biogenesis, it's going to increase the catabolic state. Well, again, to make this long story short, what we observe in this story is basically that upon infection, this high glycolytic burst is shifted towards the mitochondrial metabolism through the activation of AMPK. And actually, if we inhibit AMPK during infection, we can decrease the number of infected macrophages or the number of parasites, in vivo parasites in the spleen. So showing that, in fact, this AMPK is a protein used by the pathogen to persist inside the host. And so we identify AMPK in this context as a pro-survival, pro-pathogen survival M2 friend, let's say, protein. Because we have observed this in the beginning of the infection, don't forget we are still working on the first 24 hours, we wanted to actually look to the other side. So when we thought about MPK during this context, there was other energy sensors that are quite relevant in terms of modulating the macrophage metabolism. And two of them were E1 alpha and mTOR complex 1. Because we had access to E1 alpha knockout, we faced ourselves the question, so if AMPK is associated with susceptibility to infection, what happens of E1 alpha? It is relevant or it is not relevant during infection. So E1 alpha, as you may know, is a protein that was basically discovered on solid tumors, because it is activated by hypoxia. It is a protein that is mostly associated with increase, because it's a transcription factor. It can induce the glycolysis on the cell. It's very well described in cancers. It has been associated with the M1 polarization, so with inflammatory macrophages, and so on and so on. So this is E1 alpha, basically is controlled post-transcriptionally, because it is continuously transcribed, putting in the cytoplasm, and where it's continuously ubiquitinated by this PHD2 protein. So it's in normal conditions, without any stimulus, is continuously being produced and destroyed. On a condition that we have a stress, and here it can be deprival of oxygen, for example, it can be a pathogen, it can be other sort of stress. What happens is that these E1 alpha, this PH2 are released, are eliminated, and so E1 alpha can go to the nucleus and produce whatever the genes he has to produce, which means glycolysis, inflammation, etc. And it has been, at the time that we started studying, E1 alpha has been placed as a potential pharmacological target for infectious diseases because it is associated with inflammation. Hypothetically, if you activate T4 alpha, you can eliminate intracellular pathogens, as it is the case of Leishmania. So we and others, those time periods, we observed that in fact T4 alpha is stabilized. You can see in Western blots, it is dose-dependent stabilization of T4 alpha and time-dependent stabilization. And we observed that in fact, using this myeloid-restricted T4 alpha, so these knockouts on specific by the ESM, so on myeloid population, in opposition, complete opposition to MPK, if it deletes T4 alpha, what we have is increased susceptibility to infection. So it places, at this moment, we knew that T4 alpha was in completely opposition field of MPK, and it may be important for the control of the infection. So the absence of this factor, a little bit expected, led to decreased levels of inflammatory cytokines as TNF and IL-6. So without T4 alpha, upon infection, there is a decrease in the capacity not only to induce glycolysis, but to produce inflammatory cytokines. And actually, also, it has an increase on the immunosuppressive IL-10 that I mentioned to be one of the main immunosuppressive cytokines in this context. We try to understand why this happened. Basically, the question that we had was, okay, the activation of T4 alpha exists, glycolysis is important, but in their absence, what happens in terms of to induce that susceptibility? One of the things that we observed was that, and I'm not showing here, the increased susceptibility that we show here is actually increased through time in vitro. From day 1 to day 5, you have more and more parasites in T4 alpha knockout compared to wild type. So there is a time-dependent accumulation of the parasites during infection of this knockout. And again, making a long story short, what we observed was, was that in the absence of this T4 alpha, so we make RNA sequencing of these knockout and wild type infected by Leishmania, and we observed that in the absence of T4 alpha, we had a particular absence of this protein that is called BNIP3. So Leishmania infected macrophages, in the absence of T4 alpha, we don't have BNIP3, and actually this BNIP3 acted as a brake for mTOR activation. So what we observed in the absence of T4 alpha basically was unleashing mTOR activation, as you can see in this graphic. And actually, through several pathways, what we observe in terms of the susceptibility associated with the T4 alpha was actually not something related with the absence of glycolysis in the beginning, but actually with accumulation of lipids throughout time. So basically what we observed was absence of T4 alpha leads to mTOR accumulation. This mTOR accumulation will activate one protein that is called SRBP1C, so SRBP1C, that is leading to accumulation of lipids. And this lipid accumulation is not only observed intracellularly. So what we see here is a probe for neutral lipids. And you can see that the absence of T4 alpha, this is, I have to say, 4 to 5 days after infection, we accumulate lipids inside macrophages, but you can take a look to these images of the liver of wild type and knockout infected, and you see these lipid droplets throughout the liver of knockout animals. This was interesting to see that the absence of T4 alpha was not only related to the absence of capacity to induce glycolysis, but also to the lipid accumulation. And we try to validate this in humans. So if, as all the proteins, has a lot of polymorphisms, one of the relevant polymorphisms that we check is a polymorphism in the promoter area of the T4 alpha, and actually is a loss of function as SNP. And basically, we took monocytes from individuals with different genotypes and checked if it was according to the literature. Actually, this CTTT genotype have lower levels of FIF1 alpha compared to the CC genotype. When we infect these monocyte-derived macrophages, so these are monocytes from human donors, healthy human donors, we observed that the CT genotype was more susceptible to infection than the CC phenotype. So this, in particular, went in agreement what we observed in the murine model. So lower levels of HIF leads to higher infection capacity. And actually, I don't show it here, but we saw that this genotype in particular have much higher levels of lipids as well. So we have here a mechanism by which absence of FIF1 alpha leads to the accumulation of lipids, and by its accumulation, it's going to be associated with higher susceptibility to infection. So making this first part close, what we observe in terms of the first days of the infection is that MPK is a pro-survival parasite protein associated with an anti-inflammatory phenotype, and FIF1 alpha is the opposite, is a pro-elimination of the parasite, killing the parasite phenotype associated with a pro-inflammatory cytokines. So the role of mTOR, I'm not going to enter in many details because it is very controversial in terms of Leishmaniasis infection. We have all sorts of papers showing that, yes, a role on susceptibility and elimination of the parasites. In this particular model of FIF1 alpha, we observe that mTOR actually it is a pro-pathogen protein. So it induces susceptibility by inducing this synthesis of lipids. So coming back to the beginning, so what I've showed so far is the role of these energy sensors and these glycolysis at the beginning. Something else that we observed a few years ago that were very important is glutamine metabolism. So we looked to glutamine for two reasons. One of them is that macrophages consume a lot of glutamine, and glutamine is associated, well, some say with anti-inflammatory macrophages, but glutamine is consumed by all sorts of macrophages, being inflammatory or non-inflammatory. The way that it metabolizes glutamine can be different, but they are consumed by macrophages. And secondly, because glutamine is the main amino acid present in our serum, on our plasma, on our cells. And so PhD students of mine a few years ago, Carolina Ferreira, started to look to the glutamine. So at that moment, we had the RNA sequencing analysis. We observed that was some signature in terms of glutamine utilizing reactions. Actually, we confirmed this by qPCR, and we observed that the consumption of glutamine and the genes, the regulation of the genes associated with consumption of glutamine are time dependent. Some appears at 24 hours, some appears later on, but nevertheless, there is a generic induction of glutamine associated genes. But most importantly, we observe that glutamine is consumed in vitro. We see that infected is white, so we have much more consumed glutamine in vivo. So this shows us very clearly that, well, in the infection, the consumption of glutamine. So the next step was simple. We did several experiments. We actually went to infected mice. We didn't saw a major difference. When we saw some differences was in this case. So what we looked to infect some were treated as well, treated with methoxin. So first, one of the first line drugs against Leishmaniasis. I have supplemented the miltefosine treatment by giving glutamine. We gave a concentration of glutamine that is normally a high dose, but is a dose that is used by professional athletes, for example. And it was remarkable to see that just a supplementation of glutamine to treated mice was capable of increasing the capacity to eliminate the parasite in like 100-fold. this innate capacity provided by the supplementation of glutamine was associated with higher number of CD8 T cells, higher number of CD8 T cells that produce interferon gamma. And actually, to make this short, was that if we depleted CD8 T cells, we lose the capacity, the protective capacity induced by the glutamine. So this basically strongly suggests that there is a deficit of glutamine in the infected organ. The nutritional competition between the granuloma, where is the parasite, and CD8 T cells. As well with the drug, we can improve the effect of the drug. This can be very, very relevant to induce a faster elimination of the parasite, for example, or a complete elimination, particularly on the veterinary disease, which is more complicated to eliminate the disease. So we show that glutamine metabolism is essential. Glutamine metabolism is essential as well. If we block both, I didn't show you the data on inhibiting glutamine, but blocking glucose, blocking glutamine, sorry, blocking glycolysis, blocking glycolysis, we increase parasite survival. And as I showed you, supplementing glutamine, we can have the opposite effect. Okay, but nevertheless, we are still mostly focused on what happens in the beginning of the infection, even the data that I showed you, glutamine at first phase of infection. So we were interested in checking what happens in terms of infection at late times. For those who are not working with Leishmaniasis, that it's very difficult to have the infection going on for more than 4 or 5 days in vitro. And nevertheless, what the question was, what is happening between 1 day and 4 days post-infection? So to do so, we work in collaboration with Coral Barbas and Miguel at the University of São Paulo. And the experiment was simple. We took monocytes from humans, we differentiated macrophages, we infect these macrophages, and at different time points, we did form untargeted metabolomics. What we observed was something that was, well, expected, let's say. So when we separate all these metabolites by different types of clusters, we have all these type of responses. So some cluster that goes down like this one and then slowly recovers, others that goes up and stay up, others that goes down and stay down. Well, we have all type of motions. What we know that is dynamic as expected. And in terms of the number of modulated pathways and metabolites, we can see that from the beginning there were a lot of significant significant modulation of metabolites at 3 days post-infection. So this is reducing in time. So it is associated this capacity of the pathogen to reduce the, well, after the infection there is severe modulation, but it is capable of controlling the macrophage metabolism and functions quite well. One of the things that we observed was that most the pathways that were modulated from at this time points were associated with lipid metabolism. So we have several hits here. Metabolism, for example, linoleic acid metabolism, sphingolipid, glycerolipid metabolism. So we started to get interested in lipid metabolism because it was the very most important signature that we observed. And if you look at the heat map, we see several genes associated with several lipid pathways were also modulated. To give you 2 examples, the first one was associated with this glycerol phospholipid metabolism. So what we observed was glycerol phospholipid metabolism is associated with this phospholipase A2 that is going to metabolize the phospholipids in lysophospholipids. So it's called Lens cycle when it flows again to phospholipids. What we observe at 3, 4, 5 days post-infection is that this reaction was well, there was an accumulation of these lysophospholipids, and there is a decrease on the levels of phospholipids. So we knew that phospholipase A2 was activated, but basically this reaction is leading to the accumulation of these metabolites here. And it is very important for you to know that it's not only specific to the first days of infection. We did metabolomic analysis 1 month on the liver of infected mice, and we found the same signature with accumulation of a lot of lysophospholipids of all sorts of families. And to try to understand why this, what is the impact of this, we did a simple experiment, which was to inhibit phospholipase A2 activity. It is true that when we inhibit phospholipase A2, you inhibit accumulation of lysophospholipids, but all this pathway as well. But nevertheless, what it shows is that inhibiting phospholipase A2 activity, so inhibiting the accumulation of these lysophospholipids, we increase the number of parasites. This basically suggests and strongly suggests that this pathway is induced by the pathogen in order to restrict their own elimination. But more importantly, apart from these signatures in lipids, what we observed was something that was previously described in Leishmania, but on other pathogens as well, which was lipid droplet accumulation. So what we observe basically is this. As time goes by, there is a time-dependent, dose-dependent accumulation of lipids. So these lipid droplets basically are bags of phospholipids that contain inside triglycerides and esters of cholesterol, and they accumulate on infected macrophages, and they do not only accumulate, but also, as you can see in this electronic microscopy picture, this is a pathogen, a Leishmania, inside the phagolysosome, one and second Leishmania here. We see there is some co-localization, or at least these lipid droplets stay in the vicinity of the phagolysosome in infected cells. So because this was very clear for us, there were several hypotheses that we placed, and we are trying to answer them. One potential hypothesis is that this lipid accumulation and placing side by side with phagolysosome can lead to the feeding the pathogen. So there is a nutritional environment. So the form of the parasite inside the phagolysosome is known to be very dependent on beta oxidation of lipids. So this co-localization can serve as a feeding of the pathogen itself and nutrient and energy source. It can also protect the pathogen from these microbicidal ROS and other molecules, because these lipid droplets are quite reactive to ROS and nitric oxide species. So it can have a peroxidation of lipids very fast and protect them from killing. It alters several things, including phagocytosis, membrane fluidity, antigen presentation, and so on and so on. So what we started to and trying to do in understanding how this is done, the first thing was we knew that the lipid accumulation is time dependent. And in this graphic, we can see that it's specific to infected cells and not bystander. Actually, the accumulation of lipids through time is happening only on infected cells and not on bystander cells, cells that are on the same wall but are not infected. So it's an active process induced by the pathogen. What we know as well, it is dependent on the consumption of glucose. So if we remove the capacity of the cell to uptake glucose, we don't have this lipid accumulation. And we know now that it's dependent on the de novo synthesis of lipids. So if you block lipid synthesis, either by blocking ECC or by blocking this fatty acid synthase, as you can see, you reduce the capacity to accumulate these lipids. So basically, this tells us that this is an active process induced by the pathogen that induces the synthesis of lipids in infected macrophage dependent on glucose, and we don't know which are the stimulus from the pathogen that is going to induce this accumulation. Nevertheless, this accumulation of lipid droplets is positive for pathogen persistence. In order to explore this, what we did basically is to use a model of lipid enrichment. Basically, we differentiate monocytes to macrophages in the absence or presence of adipose tissue. So when we do it in presence of adipose tissue, we got these fat macrophages, these lipid-laden macrophages that have more or less 3 to 5-fold more lipids than classical bone marrow macrophages, that basically are a model for what is known foam cells, so in vivo. And the first thing that we observed was, so using Leishmania RFP, so the red dots are Leishmania, the green dots are natural lipids. This accumulation of lipids associates with higher parasite burden on these lipid-laden macrophages. And Marta has been exploring this. So not only this accumulation of lipids leads to higher levels of pathogens inside macrophages, but what was more importantly for us was that, in fact, this accumulation of lipids inside macrophages abolished the capacity of amphotericin B, which is as well one of the first-line drugs against the pathogen, to kill the pathogen. So this is anti-parasite activity, and when we use macrophages, lipid-laden macrophages, the capacity of amphotericin B to kill the pathogen was considerably reduced in vitro. So basically, this data shows that accumulation of lipids not only associates with the susceptibility to infection, but it dampens the capacity of the drug to kill the pathogen. Apart from that, as I showed you before, in terms of the potential mechanisms by which lipid accumulation leads to susceptibility, one of them was the protection from this killing molecules from the macrophage, ROS and other molecules. So to prove that, we did an experiment, very simple experiment, which was to take bone marrow macrophages, infected bone marrow macrophages, or infected lipid-laden macrophages, and give them SNAP. SNAP is not only, it's only a nitric oxide donor. So what happens when we give SNAP? SNAP goes intracellularly, and it's going to transform, it's going to induce nitric oxide. So by inducing nitric oxide, we expect to eliminate the pathogen. And actually, what we observed was the capacity of nitric oxide to kill the pathogen was, again, considerably reduced on these lipid-laden macrophages. So meaning that the fat macrophages have a capacity to a reduced capacity to kill the pathogen by these reactive oxygens, the nitrogen species. The second thing that we hypothesized was that lipid accumulation can interfere with antigen presentation. To try to show if it is true or not, what we did was to take macrophages infected. Again, the same type of experiment. We take bone marrow macrophages or lipid-laden macrophages, infected cells, and then place them into a culture with CD4 cells. And we measure CD69 as a marker of activation and cytokines associated with T cell activation and proliferation. And clearly, what we observed was that these lipid-laden macrophages have a reduced capacity to activate T cells and to produce inflammatory cytokines. So not only that these fat macrophages are less capable of killing the pathogen, but they're also less capable of presenting the message, the antigen message to T cells. Coming back to the in vitro, in fact, and I'm going to take just 5 to 10 minutes. What we knew was that, in fact, in vitro data show us that these cells are more susceptible. So we tried to mimic this in vivo, and to do so, we use this type of high-fat diet model. So we fed the animals with high-fat diet and compared to standard fat diet. So it's not really an obesity model, but it's more or less an overweight model. So we have more like 10% of 15% of the weight. Basically, we observe the same phenotype as in vitro. High-fat diet fed animals were more susceptible to infection, both as shown in the spleen and liver and as well in adipose tissue. I don't show here the bone marrow, but also in bone marrow we have higher parasite burden. So we have all through the mice are more susceptible. And this susceptibility associates with a less capacity to induce these Th1 cells that I mentioned previously that are important in control of the infection. So reduced capacity in producing interferon gamma producers and TNF producers CD4 T cells. Because we saw previously that lipid accumulation impedes or dampens the capacity to anti-Leishmania drugs to work, we did the same experiment in vivo. So we treat animals with amphotericin B, both on standard fat diets and high fat diets. And basically we observe the same, more or less same phenotype. The capacity to eliminate the parasite by a short time period of amphotericin B is considerably reduced. Basically 50% reduction in the spleen and in the adipose tissue, in the liver more or less the same thing as well as in the bone marrow. So this completes the idea that in fact lipid accumulation, high fat diet are associated with susceptibility and to reduce capacity to be treated. And because we look at the infection on adipose tissue, so it's not the first time that that someone observed that Leishmania can be at adipose tissue, but similar to other pathogens, namely Trypanosoma brucei and others, that goes to the adipose tissue. So we look at that. And we look basically at two types of adipose tissue. From one side, the subcutaneous adipose tissue, and on the other, different types of visceral adipose tissue, like the gonadal, omentum, or mesenteric adipose tissue. And actually, it is very interesting to see. This is unpublished data that we are still working on. It is interesting to see that on infected adipose tissue, and don't forget that we don't use the normal route of infection that is by the sunfly. So we inject parasites intravenously and intraperitoneally, so non-classical routes of infection. But nevertheless, what we observed was that, where is my place here? What we observed was that on infected adipose tissue, we have all these types of inflammation, of inflammatory infiltrates induced by mostly T cells and macrophages, and we have a considerable reduction of the adipocytes in most of these adipose tissue. This is from 1 month post-infection, so we have more data on this, showing that is something that happens throughout time. So there is a consumption of the adipocytes through time, which should be dependent on the infiltration. And something that is very interesting that we are starting to look on, that is different considering the sex of the animals. So females have a different phenotype than males in terms of this adipose tissue. So nowadays, and for those who work on adipocytes, or at least have interests on this field, so adipose tissue is more or less considered like an immune cell hub, let's say, not necessarily an organ, but a cell hub for cells. And it has been described in several pathologies, namely comparing the lean versus obese, no matter the reason of obesity, that the adipose tissue from a normal individual, lean individual, passes from an anti-inflammatory environment through a very inflammatory inflammatory environment with a lot of M1 macrophages, lymphocytes like Th17, CD8 cells. So an environment of inflammation that accumulates with increased size of adipose. And something that was interesting, making this the link to Leishmania, is that I told in the beginning that Leishmania is eliminated by Th1, by CD8 cells, by M1 macrophage. And actually, it's exactly what we have on inflammatory adipose tissue. We have a lot of inflammatory cytokines. We have M1 macrophages, a lot of Th1, Th17. Nevertheless, I show you that macrophages in this adipose tissue, fat adipose tissue, have parasites grow better than in lean tissues. So that's some incongruency that we are trying to see. What we know, in fact, in testing this high-fat diet infected animals compared to standard fat diet, is that we have an accumulation of certain amounts of cytokines. So this confirms this inflammatory phenotype that we observe on obese adipose tissue. And at the same time, we didn't detect this nitric oxide and reactive oxygen species so well. Not saying that they don't exist, but I think what happens here is that we don't detect them because they react very fast with the fats that exist in this environment. And to finish, what we want to understand at this moment is why the parasites stays in the adipose tissue, in this inflamed, overweight adipose tissue so well, how they cope with this inflammation, which supposedly it's meant to kill him, and what type of different cell types, what type of different subpopulations, habitat this adipose tissue that can be responsible for the permanence of the pathogen. I'm going to end with one of them, one specific population that we identify. Basically, it has been described, and this is a quite simplistic way to look at macrophage on the adipose tissue. Nevertheless, we can look at macrophages. So on terms of markers, F480, CD11B, double positive, of course, but in terms of the positivity for CD11C. So these resident-like macrophages are negative for CD11C, while the recruited-like macrophages, so the monocytes that comes from the blood and from the lymph that to the adipose tissue, to the inflamed adipose tissue, are CD11C positive as well. And what we observed was not only that these cells accumulate on obese adipose tissue, which was previously described, but basically are these particular macrophages are the one that accumulate much more lipids and are the one that bears more parasites. So we don't know if they bring the parasites from circulation and go into the adipose tissue, to the obese adipose tissue, accumulating lipids and serving like a reservoir, potential reservoir for the pathogen, or if they migrate to adipose tissue, get infected there and become more susceptible. That we don't know. Nevertheless, what we observed was these cells are particularly responsible for the higher parasite burden inside macrophages on the adipose tissue of obese or overweight animals. So to close, what I tried to show you in this presentation, a quick presentation, was that, well, as a dogma in immunometabolism, metabolic reprogramming is crucial for its functions. And we have been trying to dissect these cellular and molecular mechanisms. So I show you the role of glutamine, show the role of glucose, and different players like AMPK, which is pro-survival, IF1 alpha, which is pro-elimination of the pathogen. I ended to talk about lipid metabolism because, as I told you during my talk, most of the signatures that we have in terms of metabolism, metabolome, in terms of transcriptome at later time points of infection or even during in vivo infection points to a huge modulation of lipid metabolism and lower impact of glucose on glutamine. Don't forget, this is a chronic pathogen that stays in the animal or in humans for months. And so we think that understanding better what happens in terms of this interaction of lipids and pathogens and parasites can lead us to a good new immunotherapeutic strategy to help eliminating the pathogen. Let me just thank you all the people that contribute to the works that I presented here, and also the funders of all these works, and of course to you for being here, for listening, and I'm happy to take any questions that you have. Thank you. I will leave this slide.
All right, thank you very much, Ricardo. It was a great presentation. So we're open for questions. We always give preference to the students.
I will remove this.
So, are there any questions in the audience? If not, I can start with I think it's kind of it's not so philosophical, but it's kind of philosophical question too, because there is it's already old paper or a group of old papers from David Sachs' group, where they showed that Tregs and the production of IL-10, not only Tregs, but those iTregs, induced Tregs that produce IL-10, this was responsible for persistence of Leishmania major parasites in the skin, and that was important for inducing memory. So do you think in this case, the induction of lipid-full macrophages in the adipose tissue as a niche for persistent parasites can be important also for induction of memory against a reinfection in case of cure?
That's a very good question, actually. I never thought at that perspective. It should be tested, basically. What we're trying to see is that what I show you in terms of the elimination of the parasites with the drugs, they are more reluctant to be eliminated. I didn't show you all the data, but well, if you give a full dose in the spleen and the liver, you can eliminate. In the adipose tissue, you have very much difficulties in eliminating the parasite. Perhaps that persistence can be associated with a bad memory and long term. That should be, I think it's a very good experiment to be performed to check the T cell memory, later time points between high fat and low fat, and share the TCR, their affinities as well. I think it's a very good experiment.
And also a follow-up question on this, like, do you have data if in humans that were cured from Leishmaniasis, visceral Leishmaniasis, you can still detect persistent parasites in the adipose tissue in the white adipose tissue or something? Yeah.
Well, we have difficulty in getting human samples. So we are talking about visual disease. So we have very few on Europe. We try always to get from India and Bangladesh, etc. It's not as easy as cutaneous leishmaniasis. That's why we try to look at this in a veterinary setting. Look at dogs. It's much easier to get samples. We can do a follow-up throughout a kinetic analysis. It's easy to get the adipose tissue as well. And we are actually doing that with the University of, with UFBA, with the Federal de Bahia. It's an ongoing work to try to see that. And especially because dogs normally are more reluctant to be treated, not reluctant, they don't eliminate as well the pathogens as humans.
Thank you very much.
Thank you. Bruno?
All right. Thank you very much for your talk. It was very, very interesting, very nice set of data. I was interested in the lipid-laden macrophages and how they actually interfere with the Leishmania infection. And in your last figure, there is a mention of prostaglandins. And I was wondering if you actually try to see if these lipid droplets and macrophage cytoplasm were actually functioning as platforms for the synthesis of eicosanoids, particularly PGE2, that has been shown by several groups as having infection promoting effect, right?
Yeah. Actually, I didn't quantify to get you a direct response. I didn't quantify on these lipid-laden macrophages, but when we did a paper on IF1 alpha, which accumulates lipids as well, one of the questions of the reviewer was, have you checked PGE2? And we checked on IF1 alpha and it was the same. So it didn't change the levels of PGE2 compared to wild type. It doesn't mean that these lipid-laden macrophages that have much more lipids than the IF1 alpha infected cells can have produce. I think you're right. I think one of the things that susceptibility is associated, well, one of the things that contributes to susceptibility of these cells should be as well prostaglandin production as well. So on all those hypotheses that are placed, prostaglandins, I'm not going to put the presentation. but prostaglandins are there as well. I didn't mention, but are there. So it is how do you say that? Multifactorial, by which fat macrophages, let me call it that way, it's easy, are more susceptible to the infection.
Thank you.
So do you have any extra questions, guys? Anybody from Rio besides from Bruno here in São Paulo, the students?
Not on our side, I guess. Not sure if we have something in chat.
I don't see anything in chat too, do we?
Yeah. No?
If you don't have it now, you can contact me later, and you can find my email on University of Minho, and you can send me an email if you have any question or whatever you want to ask.
Oh, great. So is this a question, Fabiani? No? No. Well, anyway, I would like to thank you a lot, Dr. Ricardo Silvestre,
Actually,
Does Bruno have a
Diego, I'm sorry. No, it's just that since we have Ricardo here, we might as well ask more questions, right? So you were actually focused on the macrophages in the adipose tissue. And have you ever checked any of the other cells that were actually included in your figure? For example, the role of eosinophils or the lack of, and the neutrophils in the processes that you studied?
Actually, on innate cells, no, but I recently submitted a project to look on T cells. Because on chronicity, what I think is that this Th1 is essential. Without CD4 T cells, classical old experiments, that if you don't have Th1 cells, the mice dies, right? So CD4 is crucial. Everybody knows that. So we're trying to see what happens in terms of, well, the project is basically focusing on immunometabolism of T cells, because I think it should be associated with the capacity of these T cells to eliminate the pathogen. It should be interesting to look to macrophages and to neutrophils and other cells. Well, you know, if there is money or people who can do it to do single cell RNA sequencing on the granuloma, for example, on laser dissected granuloma, you get all these answers because you are going to check all the cells that are there. And so I believe that should be the way to try to take a look to more closely at what happens during infection, is looking at granuloma of chronic infections and looking to all types of cells. My particular perspective is on Th1 or T cells at least.
Right, and another thing that was kind of a surprise for me, because I would expect that the fat overload in the adipose tissue would drive more of M1 phenotype that is usually associated with the resistant to infection and the elimination of the parasite. So what do you think is going on there? That's or what is classically M1 environment? It's actually keeping the parasites alive.
Travou?
É só um minuto que travou aqui.
Tá bom.
Ok.
Quer responder por aqui? Não?
Can you hear me?
Yes, yes.
Yeah, okay. Your question was about adipose tissue and M1 inflammatory environments, right? So yeah, that's a surprising thing. So you should assume that it's going to eliminate the pathogen, but it doesn't. Well, we have all those hypotheses of ROS, of natural lipids acting like a shield to the pathogen, so it stops the NO and ROS, it feeds the parasite, etc., etc. It's a lot of things. Another thing that can happen is the threshold, because you are in a Yeah.
A conexão tá ruim. Agora voltou.
I'm not sure what you listen, but basically is we might think that the pathogen itself has a higher threshold for elimination. What we need to do, I think, is to look to the pathogen inside the adipose tissue. If you take a look to the work of Luisa Figueiredo from Portugal, from IMM on Trypanosoma brucei, you can see that Trypanosoma brucei, as you know, is extracellular, but it goes to the adipose tissue and it completely shifts its transcriptome to be there. So perhaps Leishmania can adapt to that environment. For example, imagine increasing trypanothione, for example, something like that, and can be fit to live in that harsh environment. That we have to look upon.
Yeah, perfect. Thank you very much. It was really, really very nice data. Congratulations. Diego, please.
I think I'll leave to Sander to do the closing this semester's immunowebinars, because this was the last seminar from this semester. So I would like to thank Ricardo a lot. It was a very nice set of data. It's very enriching for everybody here, and I'll leave it to Sandra. Thank you.
Thank you, Diego. Ricardo has a nice data about the glycolysis, glutaminolysis, and lipids. It's so amazing that I think about the Leishmaniasis. Thank you very much, Ricardo, by accepting to visit us and accepting to take this message today. Thank you very much.
Thank you. And have a nice day.
Well, thank you.